So why are we penguins? Because you're dressed for success. As penguins, you are going to be successful in AP biology. And that's why you're here in this live stream to try to help you to better understand AP biology and in the cellular energetics. So what are we doing today? Today, we're going to go over enzymes. We're going to go over cellular respiration. We're going to go over photosynthesis. We're going to go over practice questions. And then we've got just a Q&A. And so as we go through this, there seems to be a delay, about a 30-second delay between when you hear what I've already said and when you have a chance to then type it. So I've got Miss McClinton in the chat, and she's going to be my chat Q&A. So let's just go ahead and say thank you. And if you have any questions as you're going through this, go ahead and just type them in the chat and she'll help you out. And then at the very end, we'll have a Q&A where you can ask any questions that aren't answered yet, or you have that chance to be able to ask any extra questions that you've come up with. So first thing, gives free energy and the reactions that take place with it, right? So first thing, what is gives free energy? Why do I even care about it? So this is the energy available to do work. And so there's this really complicated equation that actually isn't on the formula sheet anymore, but it's understood that you already know this information. And so you're going to be able to apply the information from this formula because you already took chemistry and you already took regular biology. So real quick, I want to go over what this equation is and what it all means. So the delta G is your change in gives free energy. So it's how much energy has been released or absorbed in the reaction. The delta H is looking at your change in enthalpy. That's your heat energy. So as we know, when things heat up, they start to move faster. And when things slow down, when things cool down, they start to move slower. And so the enthalpy is just looking at that heat energy. The T is our temperature in Kelvin. And then the delta S is going to be looking at our entropy or the disorder, kind of the randomness that you might be seeing in a structure, right? So if something is really kind of structured, do all these bonds, it's going to have a low amount of disorder. But when we think about like carbon dioxide and water, and as you've broken down glucose into all these molecules, now we've increased that disorder. Now we have a higher amount of entropy in the system. So what you need to worry about is this equation right here. This delta G equals delta GF minus delta GI. So that's talking about how much of our free energy has changed in the reaction. So there's two main reactions that we're going to see here. You're going to have an endergonic reaction and you'll have an exergonic reaction. With an endergonic reaction, this means that we're going to have to absorb energy, okay? So as you see right here, this is our delta G. Our reactants is down here with its gives free energy, and our products is up here, which means that it had to absorb energy in this process. So when it created its bonds, when it formed this new compound, this is causing there to be more energy stored in that molecule. So this is our delta G, and we have absorbed energy. And so it's a positive delta G, which means that the reaction is not spontaneous. And yes, this does seem a little bit like AP chemistry. And so an example of this would be ADP plus inorganic phosphate giving you ATP. So as we phosphorylate that ADP making ATP, that's going to be an endergonic reaction. It required input of energy in order to do that. And if you think about cellular respiration, which we'll talk about in a little bit, when we're having the protons moving across that concentration gradient down, right, it's going to be turning ATP synthase to provide that energy, that potential energy to allow for that bonding to take place. Okay. So the other reaction we have is an exergonic reaction. It's going to be basically the opposite. In an exergonic reaction, we're releasing the energy. And so as you see, the reactants have a higher amount of energy than our products, which means that we have a negative delta G and we've released energy. We've lost energy. And so an example of this is going to be when ATP is broken down into ADP and inorganic phosphate. So you're oftentimes going to see these two reactions taking place at the same time in energy coupling. Okay, so as I said before, with cellular respiration, there's going to be a potential energy. That potential energy from our protons, they're going to flow down their concentration gradient. That's going to be exergonic. It's releasing energy. Okay, and the energonic reaction is going to be ATP synthase adding the phosphate to ADP. So you're going to see that they're going to be coupled together. The same thing as if we talk about active transport, right? In active transport, we talked about last week, you had the protein gets phosphorylated. You add a phosphate to it. So when ATP breaks down to ADP and inorganic phosphate, it released energy. That released energy allowed you to pump something against this concentration gradient, or the endergonic reaction. If you need a way to help to remember this, Energonic has that En, meaning that things are going to enter, right? We're going to enter some type of energy. It gets added in, right? Versus exergonic, Ex, exiting, okay? So things are going to exit and be sent out, okay? So hope that was good. Now we're going to move into enzymes, which enzymes are really important because of the fact that they're going to be applied in all the steps of respiration and all the steps of photosynthesis. So that's why we see the enzymes here. But first thing, what is an enzyme? Like, what is the function of it? So it's a biological catalyst, and a catalyst just speeds up a chemical reaction. The way that it does this is by changing the activation energy. So we didn't mention it before, but this little amount of energy right here, from where you go to the reactants, and then before it becomes kind of the product, we see this little hump. This is our activation energy. So it's the amount of energy that has to be added in order for the reaction to start taking place, okay? So like, if we're trying to weaken the bond, so we have this structure and I want to break that structure. So I'm going to have this weakening of the bonds by increasing the energy to kind of pull in whatnot and kind of strain those bonds. Or thinking about if we're trying to put two things together, okay? So all that's going to require some type of activation energy. So what enzymes do is they decrease that activation energy. So you see that we have a less amount of activation energy required in order for this reaction to take place, which means that we can go through this reaction more frequently. So the enzyme is going to speed up the chemical reaction. It's going to increase how many times we can go through that reaction. So important things about enzymes, they're proteins. So everything that we talked about in Unit 1 review over proteins applies to enzymes, okay? All enzymes are proteins, but not all proteins are enzymes. So also, they're not consumed by the reaction. They are workhorses. They will continue to work over and over and over and over and over. You can use them forever, or as I said in the Sandlot, forever. So they're not ever used up. They can keep on working. And the reason why is because they bond, they release. They bond, they release. And so they're never actually used up. The only way that you can get rid of an enzyme is to denature it. And so we'll talk about denaturation in just a couple moments. But that's the only way to stop an enzyme. And also note that it's only affecting the activation energy. This delta G does not change. So it can't make a non-spontaneous reaction into a spontaneous reaction, and it can't make a spontaneous into non-spontaneous. So it's not going to affect that delta G. And I've seen questions where they've asked you about, oh, if I've doubled the enzyme, what happens to my delta G? And the answer is your delta G is going to stay the exact same because the enzyme does not affect your delta G. It does not change that change in Gibbs free energy. So how does enzymes work? How do they work? So they've got this little active site right here, this part where the substrate is going to bind. So the substrate binds within that active site. And we know that if you bind anything to a protein, the first thing it does is it changes shape. So you're going to see that it changes shape as an enzyme binds to our substrate. We call this induced fit. I also call it a conformational shape change. So what happens there is that's now allowing for the straining of the bonds that also can allow for two things that are going to bond to now be attached appropriately. So in nature, what's happening is these molecules are all floating around. They're kind of, hopefully, they bind into one another. But the probability of them hitting one another at the exact angle that they need to hit is a lot lower. But if I can bind these two molecules and I can orient them appropriately, I can ensure that they bind together with no problems. And so that's another thing that enzyme is doing. So after it does that, we convert it into our products, and then those products are released. And now that enzyme is available to take a new substrate. Okay, so that's pretty cool. But what happens if something else binds? So we have these things called inhibitors. Now there's two types of inhibitors. You can have competitive inhibitors, which are going to bind to the exact same active site. So as you notice here, our competitive inhibitor is going to have the exact same shape as our substrate. So those two are going to bind to that active site, okay, which means that they're now competing for that site, which is why we see competitive inhibition, because they're competing for that active site right there. So how can I overcome this? To overcome this, I can increase the amount of substrate. If I increase the amount of things that are reacting, there's a higher probability that substrate is going to bind into that active site. So I can increase the reaction rate by decreasing the amount of inhibition. So what's the other type of inhibition? We can have a non-competitive inhibitor. A non-competitive inhibitor is going to bind to an allosteric site. It's another site on the enzyme. It's some other location. And when it binds that location, because we're looking at a protein, it changes shape. And when it changes shape, now this active site is no longer going to be able to bind to the substrate. Now, either one of these inhibitors can be either reversible or irreversible. If they're reversible, it means that as soon as it comes off, that you're then able to get another thing to bind. So you're able to get that substrate to still bind into that enzyme. If we're talking about irreversible, it means that it's going to bind and it's going to cause issues and not be able to bind, not be able to allow for the reaction to take place. If I remember correctly, cyanide would be considered a irreversible. It's going to bind to the cytochromes within the electron transport chain to inhibit the binding of those electrons so that we won't see the reaction take place. And so back to denaturation. So in denaturation, what happens is that we're going to cause an unraveling of our protein, right? So enzymes are proteins. And so if we see that secondary, tertiary, or quaternary structure break any of its bonds and it unravels, that's called denaturation. So let's think for a second about this morning. You probably had some eggs, or at least I'm going to hope you had some eggs. So you had fried eggs. Right. And so when you first cracked that egg on that frying pan, you saw this colorless kind of jelly substance around that yolk. Right. And then as soon as the heat increased, you then saw that colorless jelly stuff become a white solid. That's because the enzyme was denatured. I think it's ovalbumin or something. And so that protein got denatured and now it's a new structure. And so that's actually not going to be able to go back. It's going to be forever denatured. But there are some things that will be able to refold. So like if your hair is curly and you use heat to straighten your hair, when you go back into the shower or you're around humidity, it then coils right back up. So that would be considered kind of like a reversible way that you could bond your protein. So what I was going all that for was to give you different things that we have. So our environmental temperatures, if it gets too hot, it's going to cause denaturation. If it gets too cold, it's just going to slow down the reaction. It's not going to occur as fast. And then pH changes. pH changes are going to, no matter what, if it gets too low pH or too high pH, is going to cause denaturation of that protein. And these top two are the ones that are directly out of the CED. You need to know temperature and you need to know pH. An example of the pH has to do with, think about your milk, right? So you let your milk go sour. Why does your milk go sour? Because you have bacteria in the milk and the bacteria is feeding off of the lactose. And so as it breaks down that lactose, it goes through, of course, fermentation, which we'll talk about in a little bit, and that produces lactic acid. So it's going to decrease the pH of the milk. And as it decreases the pH of that milk, you're then going to start to get those curds and the nasty, lovely stuff that helps us to make cheese and cottage cheese. Yay. And then another thing that can denature is salinity. So the way that salinity affects it is that it's going to have, so salt has positive and negative charges on it. And so those polarities can mess with the polarity of the protein, causing it to denature. Whew, that is all on our enzymes. I do see that there is a question in the chat, and they're asking about, does allosteric inhibition equal non-competitive inhibition? Allosteric just means that it binds to another location. So I guess you could probably say allosteric inhibition is considered the same as non-competitive because allosteric is just another location on the enzyme. And then I think there was another question that I saw. I don't see where the question is. Oh, competitive inhibition can be, nope, I don't see the question. I'm sorry, okay. So on to cell respiration, which is why you came to this, right? So cell respiration is tough. It's real hard, right? And the reason why it's tough is because there's a lot of parts to it. So I want you to just take a moment, just take a breath. It's not needed to be that complicated. You as a student don't need to know the 10 steps of glycolysis. You don't need to know the 8 steps of Krebs Cycle. You have to have a general understanding of what's happening in the process, what goes in, what comes out, and why is it important. You don't need to know all of the itty bitty little teeny tiny little details that I had to learn in biochem when I was my bio major. So just take a moment to just breathe, because it's not as complicated as we all make it to be, right? So there are 3 different steps that we see in cellular respiration. First step, glycolysis. Second step is Krebs cycle. And the third step is oxidative phosphorylation. So glycolysis is going to take place in the cytosol. What this means is that any organism can go through glycolysis. So our bacteria that don't have a mitochondria are able to go through this. And so it's actually evolutionary believed that glycolysis was the first process that took place because it didn't need a mitochondria. We didn't have membrane-bound organelles. There was no oxygen in the atmosphere. So all organisms were able to produce ATP by going through glycolysis. So we believe that that was the first process that even took place evolutionarily in terms of making ATP. So here is the gist of glycolysis. It starts out with glucose. It then goes through this reaction. As I said, there's 10 steps, don't need to know them. And in those 10 steps, you're going to make 2 NADHs, you'll make 2 ATPs, and you'll make 2 pyruvates. So as I tell my students, 2, 2, 2. So you're like, what are you talking about the show? Anyway, so NADH, I tell my students a lie. That H stands for holding. When you break down that glucose, you're going to kind of release these different high energy electrons. Okay. So those high energy electrons have to go somewhere. Something has to hold them in order for us to get them to a process that will be able to use those high energy electrons to store that energy. Okay. So the H stands for holding. As we oxidize glucose, it's going to allow us to reduce NAD plus into NADH. So it gains an electron. In case you don't remember, oxidation is the loss of electrons. Reduction is the gain of electrons, or oil rig, as your chemistry teacher probably told you. And so we oxidize that glucose, and we are taking those electrons out of it as we break it down into these 2 pyruvates. You're going to notice there's no carbon dioxide at this point. Why? Because of the fact that we have 6 carbons in glucose, and there's 3 carbons in pyruvate. So we still have 6 carbons between these 2 molecules. So there's been no carbon dioxide released at this point. All that we've done is taken glucose and split it. Hence the name, glycolysis. Glyco meaning sugar, lysis meaning to break. All I did was break my sugar, pop. So the H stands for holding. Yes, there's a hydrogen that's released, and the hydrogen does deal with the electrons, but we really kind of more focusing on that NADH is a vehicle to hold those electrons. And as Ms. McClendon mentioned, there's actually 2 ATPs that are put into the reaction. It's called the energy investment, and then there's 4 ATPs that come out, which is the energy payoff. But you just have to, again, big picture. You just need to know there's 2 ATPs that are made in this step. Okay, so where was I at? Okay, so after we go through glycolysis, there's another process. It's called pyruvate oxidation. As we said, we're losing electrons. So the pyruvate, we said, has 3 carbons to it. Okay, in pyruvate oxidation, your teacher may call it the link reaction. Your teacher may call it the intermediate reaction. It honestly does not matter what you call it. College Board calls it the pyruvate oxidation. But again, just be able to recognize what's happening. We're oxidizing pyruvate. So our 3-carbon pyruvate is going to become a 2-carbon acetyl-CoA. And so as you see, we go from 3 carbons to 2 carbons. So you can tell me how many carbon dioxides are released. Well, that would be 1 carbon dioxide is released per pyruvate. So we're going to lose that carbon dioxide. We also see that each one is going to have an NADH that comes out. And then we're also going to see that we add this coenzyme A. Coenzyme A is just designed as a placeholder. It's going to hold the bond. So we'll be able to move it into that Krebs cycle. So it's kind of that link, that intermediate. I don't actually have it on the slide because it's that kind of, it's an intermediate, it's in the middle. So that allows us to get into our Krebs cycle. Whew, this is a complicated little thing, right? Whew. Again, we just need to know what goes in, what comes out, why is it important? So here you see we have acetyl-CoA going in, okay, and the coenzyme immediately comes off. All that we're doing is adding 2 carbons into the cycle. It's a cycle. So what you start with, you have to end with. Since I've added 2 carbons in, I have to have 2 carbons come out in the form of carbon dioxide. So you're going to see there's 1 carbon dioxide right here, and there's 1 carbon dioxide right here. These carbons have come out in the form of carbon dioxide, okay? So We also are going to see that there's 3 NADHs. We're breaking down glucose. Glucose is done after this. We're gone. There's no more glucose after this process, okay? All that's going to remain are those high energy electrons. So as we break it down, again, we're taking those electrons off, those high energy electrons. So I make NADH, 1, 2, 3, and I make 1 FADH2. Okay, so this is just like NADH, but it's a little weaker. And when I say weaker, it just doesn't end up as high on the electron transport chain, and we'll talk about that in just a second. And then I'm going to have 1 ATP made. Now, this cycle is based on 1 pyruvate. We made 2 pyruvates, so you go through the cycle twice. So if you're thinking about per glucose, that would be 6 NADHs. I'm not pressing any of these buttons, I'm sorry, y'all. That would be 6 NADHs. That would be 4 carbon dioxides, that'd be 2 ATPs, and that'd be 2 FADH2. Okay. Now this process takes place in the matrix. So if you remember from unit 2's review, and we talked about the parts of the cell, we talked about the mitochondria and how mitochondria has a double membrane. There's this outer membrane, and then there's a cristae, that inner membrane on the inside. And on the inside of the inner membrane is that matrix. That's what's taking place in there, is this process of the Krebs cycle. You have to have oxygen in order to go through this process. Number 1, there is a transport protein that brings the pyruvate into the mitochondria. Number 2, there's no way to regenerate the NAD plus if you can't lose that hydrogen. And so the electron transport chain, as we talk about in a second, is going to allow us to lose that hydrogen. It's going to allow us to lose those electrons. And so once you get into the mitochondria, you're not going to be able to regenerate. There's a question. That's why I'm answering it like that. Okay, so last step, oxidative phosphorylation. Nothing's left of glucose. Glucose is gone. There's nothing left. All that we have are these electrons, right? So we have the NADH and the FADH2, more or less, we're looking at the electrons being added to this process. And then what are the products? We're making ATP. You don't need to know the specific number. That number varies depending on a lot of different components dealing with how many protons are pumped across that membrane, okay? This takes place in a mitochondrial cristae. Cristae is highly folded. Why is it highly folded? Because there's an increase in surface area. So you can have more of these components of the electron transport chain and chemiosmosis within that membrane to allow us to make as much ATP as possible. So that's why we have a high surface area to volume ratio on that inner membrane. So there's 2 parts. You have the electron transport chain and you have chemiosmosis. The electron transport chain is exactly what we're saying. Electron is moving down the chain. Like literally, all we're doing is we go through reduction, oxidation, and they just pass it from one to the next. As they pass it, there's energy release. It's slowly dropping in energy. And as it slowly drops in energy, that energy doesn't just go away. It's used to pump a proton across the membrane. So we pump the protons across the membrane. See, look right here. You see that we have this transport protein, and we're going to pump those protons across the membrane. As they're pumped across the membrane, now, of course, we're getting this high concentration of them over here. Yay! That high concentration, that is potential energy, the potential energy that we're going to use. The electron transport chain, do you see any ATPs here? Nope, because the electron transport chain itself doesn't make any ATPs. All it's doing is allowing us to make that proton gradient. That is the only thing it's doing, is it's just making a proton gradient. It is taking those protons and pumping them into the IM space, which is the intermembrane space, just pumping them in there. Now, they've had questions about this, about the pH, right? So if a higher amount of protons in the IM space, that means that the pH of this area is lower. I have a low amount of pH because there's a high amount of protons in that area, okay? As we said, generates the proton gradient. And at the very end of this, what takes those electrons? Oxygen. So if you don't have oxygen, or you have something that's inhibiting that oxygen from binding to those electrons, this whole process gets backed up. If that whole process gets backed up, you cannot drop off any more electrons, which is a problem because you can't reduce the NADH. If you can't oxidize the NADH, then you can't break down any more glucose because you don't have NAD plus. That's a big problem that we're going to come back to in a second. And then the second part of it is chemiosmosis. Chemiosmosis is using that proton gradient. It's going to flow down its concentration gradient, comes right back again, right into the matrix, through this thing called ATP synthase. Kind of looks like a rotor. And basically it's binding, and as it spins, it's going to then use that energy of that proton gradient to phosphorylate ADP, making ATP. So the whole job of this is to make those ATPs. So together, oxidative phosphorylation. I use oxidation to phosphorylate. Easy peasy lemon squeezy. This does produce heat. There's a specific type of respiration that's mentioned in the CED. It happens in your brown fat cells, in which you can generate your heat without generating ATP. There was something about it where it's just generating the heat. It has to do with the cyclic process within the cell. Okay, so I think we've got all those questions in here. I think Ms. McClinton is answering most of y'all's questions. If not, you're welcome to ask them at the very end. Where's electron transport chain is going to be found in the mitochondrial cristae. So mitochondria has a double membrane. Here's the outer membrane, and the inner membrane is the like highly folded inner part. It's on that is where you see the electron transport chain. So I think we're ready to move on to photosynthesis. Oh, I'm pressing the wrong button. Sorry, y'all. Right, so, I'm ahead of myself. Sorry, y'all. So this question was on, I believe, the 2014, maybe 2015 exam. And so this was to try to help ease your mind, right? This is what was given to the students. You don't need to know the steps of glycolysis. You don't need to know the steps of Krebs cycle, and you don't need to know electron transport chain. You need to just have a basic understanding. So you were describing a contribution of each of those in ATP synthase. As I said, what goes in, what comes out, why is it important? So they asked you, why is this important? How does this make ATP? So it makes it because it makes NADH to go to electron transport chain. It makes it because of the fact that I'm now making acetyl-CoA to go into Krebs cycle. And substrate level phosphorylation. Again, substrate level phosphorylation makes NADH for electron transport chain. And it's transferring those higher D electrons to the electron transport chain. All that is important. Plus, again, GTP making the ATP in our substrate level phosphorylation. And then last but not least, how does this make ATP? Creates proton gradient to allow for us to synthesize ATP using ATP synthase. So again, this was meant to just kind of help to ease your mind. The blue parts are the questions. And then this down here was the scoring guideline that they gave you. Can I say it also occurs in the inner membrane, or is the cristae the correct answer? You could say inner membrane, or you could say cristae. Either one of those answers are interchangeable. It honestly doesn't matter. We, as the readers in your AP exam, knows what you're talking about. But you should know that the inner membrane is called the cristae, because if there is a multiple choice question and they're describing the cristae, they're talking about the cristae, then there's going to be you don't know what they're talking about. And that has come up before. Specifically this question, teachers were upset because of the fact that the students didn't know this as the Krebs cycle. They knew this as the citric acid cycle. And so a lot of students weren't able to answer the questions because they didn't have any understanding of what this was. So do understand that there are some terms that are interchangeable. So on to photosynthesis. All right, so there are two steps to photosynthesis. Same thing, just have a basic understanding of what is going on. You do not need to know all the itty bitty little itty bitty steps, but you have a basic understanding. So we have the light reactions, and we have the Calvin cycle. So this is the thylakoid membrane. In the thylakoid membrane, you have these different little photosystems. Okay, so this right here is photosystem 2, and this is photosystem 1. Yes, it's crazy that they're named in opposite orders, but they're named based on when they were found. They first discovered photosystem 1, then they discovered photosystem 2, and it's very hard to go back into the literature and change things, so they just made it a new name. So what's happening here is this is an electron transport chain, right? So light reaction needs light. Light is going to shine in on the photosystem. What happens is there's these little pigments, chlorophyll, that's going to absorb that energy. As it absorbs that energy, our electrons jump to higher energy levels. The only way that you can lose energy is, number one, it falls back down by releasing it as heat, release it as light, or transferred it to another pigment. And so you're going to see this like bouncing around of these antenna pigments, bringing the energy to our reaction center complex. That then, here's that P680, that's reaction center complex. It then gets stolen by a primary electron acceptor, and then it goes down the electron transport chain. So just like what we saw in mitochondria with electron transport chain, the same thing happens here. That electron has a high amount of energy that it gained from the light, from the solar radiation, okay? And so as it's falling down in energy, it's creating a proton gradient. So here you see the protons being pumped across the membrane, okay? The way it's pumping it is just pumping it into the thylakoid space. And so then moves into photosystem 1, same idea. It's then going to go in the electron transfer chain to then go into NADPH. Wait a minute. Did you just say what? What? So NADH is in cellular respiration. NADPH is in photosynthesis. They both are going to carry electrons to the next part. Okay, they're both doing that. The pH is only seen in photosynthesis. The NADH is only seen in cellular respiration. Do not get tripped up on that, because that is a way that they can ask you a question, a multiple choice question, and you could select the wrong answer because you didn't make that connection. You didn't see that. So do make sure you know NADPH is in photosynthesis. And so this will carry the electrons to our next part, okay? And then we can make ATP using ATP synthase, allowing those protons to move through, okay? So as I said, where does it take place? In the thylakoid membrane. It starts out with water. We didn't talk about water. Water's right here, okay? So what happens is that the water is broken. It's called photolysis. It breaks water using light, break. It's going to go through photolysis to break that water molecule, which is going to regenerate those electrons. We said that these electrons get lost by the electron transfer chain. So you can't always lose your electrons. So the electrons that are coming from water kind of replace the ones that were lost. We also have photons. So the light energy coming down that's shining on our photosystem. Okay. And what do we make? We make ATP and NADPH. Now there's two different types of electron flow. What I just described was linear electron flow. Okay. It goes from photosystem 2, goes down this electron transport chain, photosystem 1, and then it's absorbed by NADPH, and we make ATP. Okay. So, and the process right here is what is allowing us to be able to make that ATP. So kind of some people think about this is where the ATP is made, even though it's technically over here, but whatever. So you use photosystem 1, photosystem 2, and you make ATP and NADPH. Cyclic electron flow has to do with only using photosystem 1. When the electron is stolen, it goes back down this electron transport chain. And so it literally has the exact same electron going in a circle, which is why we call it a cyclic electron flow. It literally just keeps going around in a little bitty circle. Why does it do this? Because I need to make ATP. There is an unequal amount of ATP and NADPH needed for the next step in the Calvin cycle. Unequal amount. And so the only way for us to not have all of our NADPHs held up is if we can make ATP alone. So some of your photosystems are already built up that they're going to be cyclic. And some of them are already set up so that they're going to go through linear. Between both of them, we're going to make enough ATP and NADPH to be able to fuel into the Calvin cycle. So I think I said all that. I did not mention about the which direction. So when we looked at cellular respiration, we talked about that the protons were pumped into the ion space. A difference here, now they're pumped into the thylakoid space. So kind of thinking about that things were pumped out before, now they're being pumped in. The goal is to put it into a small location to build up that concentration. So again, where is there a low amount of pH? Inside the thylakoid space, because there's a high amount of protons in there. So Calvin cycle. Whew. How do you have another cycle? All these cycles, and they're so confusing. So sometimes you need to find ways to remember. So Krebs cycle is going to be found in cellular respiration. And then Calvin cycle is going to be found in photosynthesis. You can kind of come up with some type of mnemonic if you want to try to keep those together. You know, you're not going to put the two C's together. So CK and then remember PC. It could be a way to remember. It's up to you. So what's happening here? Carbon dioxide goes in. It's going to go through this cycle and it's going to make G3P. Okay. So where does it take place? In the stroma. The stroma is the jelly substance that like cytosol of the chloroplast. Okay. And so we're pumping, of course, we've got the, not pumping, sorry. We've got the ATP and the NADPH that's being synthesized, you know, all that fun stuff. It immediately goes into that stroma. And so it's going to be able to go into the Calvin cycle. Carbon dioxide is coming in through the pores of our leaf. called our stomata. Yes, stroma, stomata, very similar. So it comes in through the pores called the stomata. So the carbon dioxide is then going to bind to the RUBP using the enzyme of Ribisco. Yes, those words are important. So carbon dioxide binds to RUBP using the enzyme Ribisco. The rest of the stuff isn't as important. We're just rearranging and we're reducing, regenerating, rearranging. There's just all this stuff happening, okay? The big thing to understand is that we are going to use the NADPH and we're going to use the ATP. Over here is a 1-to-1. We have 6 NADPHs and 6 ATPs. Over here, we need an extra ATP. That's why we need the cyclic electron flow. So starting material, we have 3 carbon dioxides. I have 9 ATPs, 6 NADPHs, because I need to have 1 NADPH and 1 ATP per molecule in the Calvin cycle. It kind of has to do with something, don't worry about it. And we're going to make 1 G3P. Why is there only 3 carbon dioxides? Because G3P only has 3 carbons. You have to put 2 G3Ps together in order to make glucose. And then the product, G3P. So I think that we've gotten our photosynthesis down. Does photolysis, during photolysis, does the proton resulting contribute to proton gradient? Yeah, that's the protons that end up getting pumped, is that they have those protons to be able to pump them. Same thing as when you look at the cellular respiration. The water right here that's formed when the oxygen takes the electrons has to do with those protons that we do have being pumped across that membrane. So that brings us to the end of the lecture component of this. Now let's talk about some multi-choice practice. So chemical reactions for photosynthesis. They give us a reaction. I don't have to memorize. Amazing. So here we see 6 CO2 plus 12 waters plus light energy gives us glucose plus 6 oxygens plus 6 water molecules. And so they say that if the input of water is labeled with radioactive isotope of oxygen. So what they said is the oxygen, right? We're going to put a radioactive label. It's a oxygen 18. It's got, if you remember about our isotopes, it means that they have a different neutron number. So they weigh a little bit more, but they have the exact same number of protons and electrons as the other isotopes of it. And so it allows them to track it. So they want to know the oxygen gas that's released. So this oxygen right here, where does it come from? Because they're finding that it has the oxygen 18 in it. Where did it come from? How did we get it? So they want to know what's the most likely explanation. So during the light reaction of photosynthesis, water is split. Yeah, that happened. Awesome, cool. The hydrogen atoms combine with the carbon dioxide. No, we didn't talk about that happening. And oxygen gas is released. That did happen. Okay, cool. So we have something that's wrong in this. And you're going to see that happening a lot, where there's some stuff that's right in your answer choice, but there's also some stuff that's wrong. So you need to look very closely at your answers to ensure that you're picking the right one. So this is a strategy. You can make little check marks and X marks to try to help yourself. You can write in the exam book. So make as many marks as you need to on your test. During the light reaction of photosynthesis, water is split. Awesome. We're good. We got the right answers. Removing electrons and protons. Cool. And the oxygen gas is released. That is all true statements, right? So let's just keep going through our answer choices, make sure that we didn't make an error, right? So during the Calvin cycle, water split. That didn't happen. Regenerating NADPH from NAD+. That didn't happen. We may. Let it know. And then the oxygen gas is released. No. So during the Calvin cycle, water split. Nope. Hydrogen atoms are added to the sugar. No. Oxygen gas is released. Nope. So we know the only logical answer we have here is B, right? During light reaction photosynthesis, water gets split. We talked about that. Photolysis. It's in photosystem 2. We split the water. It releases those electrons so that the photosystem 2 will get its electrons back, which is going to end up releasing those hydrogens, making our oxygen. So we'll be able to go through our process. Woo! So next one, of course, I just did a photosynthesis question. So I wanted to make sure that we also go through a cell respiration question. So an experiment to measure the rate of respiration in crickets and mice at 10 degrees Celsius and 25 degrees was performed using a respirometer. You probably did a very similar reaction in your class in which you were looking at different P's, an apparatus that measures changes in gas volume. Respiration was measured in milliliters of oxygen consumed per gram of organism over several 5-minute trials, and the following data was obtained. So it's talking about respiration. It is talking about oxygen. So how does respiration and oxygen like work together? So if you're going through more cellular respiration, right? If you're going through more of it, you're going to be consuming more oxygen. Why? Oxygen was that final electron acceptor. So if you have more respiration, you're going to use more oxygen to get all those electrons at the end. Okay. So there's a connection there. Crickets are ectotherms. They're cold-blooded organisms. They do not require any type of like cellular respiration to keep themselves warm. If they're cold, they move out in the sun and they bask in the sun. They get all this solar, thermal energy. Versus a mouse is an endotherm. It has to make itself warm, you and I, right? And so it goes through more cellular respiration if it's cold than when it's warm, because of the fact that it has to make all that heat as a byproduct of cellular respiration. So here we see the data, right? So we've got 10 degrees Celsius, 25 degrees Celsius. We've got the amount of respiration. This is according to the data. The mice at 10 degrees Celsius demonstrated greater oxygen consumption, okay, cool, per gram of tissue than did the mice at 25 degrees Celsius. This is most likely explained by which of the following statements? Cool, okay. The mice at 10 degrees Celsius had a higher rate of ATP production than mice at 25 degrees. Well, I'm going through more respiration, I'm using more oxygen, and I'm making more ATP. That sounds plausible. Okay, cool. The mice at 10 degrees had a lower metabolic rate than the mice at 25 degrees. Well, if it was lower metabolism, I wouldn't be having more oxygen consumed. So B's out. The mice at 25 degrees Celsius weighed less than the mice at 10 degrees. That doesn't matter because it says that we're looking at per gram of tissue. So I've already standardized it so that we're looking at the same gram per tissue. So it doesn't really matter how much the mice weigh. The mice at 25 degrees were more active than the mice at 10 degrees. Well, if they were more active, they would go through respiration more frequently, and we would see a higher rate of oxygen consumption by those warmer mice. So the only logical answer is A. Whew, that brought us through our multiple choice. Let's get into some free response. So this question is from the 2021 exam. It's actually number 3. So I wanted to let y'all see different examples of FRQs. Last time, I think we looked at an example of a number 5. This week, we're looking at an example of a number 3. So researchers hypothesized a plant compound, resveratrol, whatever that says, improves mitochondrial function. To test this hypothesis, researchers dissolved the R word in dimethyl sulfur oxide, or DMSO, which our chemistry people always use it so much. I love that chemical. The solution readily passes through cell membranes. They add the resveratrol solution to the mammalian muscle cells, drawing in nutrient-rich solution that contains glucose. They measure ATP production at several time points after the addition of the solution and find an increase of ATP production by muscle cells. Whew, that's my prompt. So I need to describe the primary advantage of a muscle cell using aerobic respiration over fermentation. Wait a minute. We never talked about fermentation earlier. I'm sorry, y'all. So fermentation happens after glycolysis, okay? The whole point is that you have no oxygen, and so you can't go through the rest of cell respiration. You can't go through Krebs cycle, and you can't go through oxidative phosphorylation. So what they do is you go through fermentation, making lactic acid or ethanol, and it allows you to regenerate the NAD+, so you'll be able to go through glycolysis again. So your cell goes through glycolysis, then it goes through fermentation, and that's all it basically does to bounce the NADH and the NAD+ back and forth. So describe the advantage. Well, the advantage would be that if I can go through all of cell respiration, I can make more ATP because I only made 2 ATP molecules in glycolysis. So fermentation only makes 2 versus if I go through all of aerobic respiration, I make the 2 in glycolysis, I make 2 in Krebs cycle, and then I make, I think it's like 34 to 36 in the oxidative phosphorylation. So I make about 36 to 38 molecules of ATP by going through aerobic respiration. So more ATP per glucose is produced by aerobic respiration. That is the advantage. The second question, identify negative control. Wait a minute. That's just has to do with the experiment. Exactly. So some of these questions on the exam isn't based on your biology knowledge. It's based on also science practices and whether you can apply those science practices. So coming up with a control, coming up with a hypothesis, coming up with a null hypothesis, those are all parts of your scientific processes. and your scientific practices. So appropriate negative control for the experiment that would allow for the researcher to conclude that ATP is produced in response to the treatment, right? So originally we had this dissolved in DMSO. If I want to make sure that it is the treatment that affected it, then it has to do with the fact that I need to compare it to something that doesn't have it, right? So the researcher will run the experiment without adding the chemical, or they treat the cells with DMSO and only DMSO. So that would allow us to ensure that it is the chemical treatment and only the chemical treatment that caused this effect in our cells. Number 3, predict the effect of short-term ATP production when treated mammalian cells are grown in a culture or medium that lacks glucose or other sugars. So I have no glucose and I have no sugar. Wait, if I don't have any of those things, can I make ATP? Well, no. So you're going to find that there's no ATP. Another answer you could have given was reduce ATP. You can actually break down your fats and you can break down your proteins. They added different portions of respiration. Your fats will be added into the Krebs cycle, and then your proteins, I believe, are added into the Krebs cycle. And I think there's another, like they add into like the intermediate, I believe. So you can still break down the fats and you can still break down your proteins to be able to synthesize some ATP, but it's a reduced amount of ATP. So those two were options you could have put here. And the last thing we had, researchers find that the chemical stimulates the production of components of the electron transport chain. Researchers claim that the treatment with the chemical will increase oxygen consumption by the cell if glucose is not limiting. Justify your claim. So they're telling you something. They said that if I use this chemical that is going to work in the process of mitochondria and improve the function of the mitochondria, that I will see an increase of oxygen. Well, why will you see an increase of oxygen? Because oxygen is a vital electron acceptor. So if I'm going through more processes within my mitochondria, I'm using more oxygen. So that was that last question. So I have this slide up. It says the Q&A. So there's a 30-second delay between what I'm saying and what you're hearing. And then you have that time to type it into the chat. So I'm going to show you something real fast. And while I'm showing you that, you go ahead and type up whatever questions you have into the chat so that we can talk together. Okay, so number 1, let me show you this website. So APBioPenguins.weebly.com. Okay, so if you go here and you go into Marker Learning Sessions, you can actually access this whole PowerPoint. That way you can, it's right here. It'll be download, and I've got the previous ones. You can still access all of those as well. And I'll keep updating this website. Okay, they'll have those. So you have able to grab that. I make these little, I take all the PowerPoints and put them together as like a 1-pager review. So you can just print out that 1 page that has all that information. Also, you can see that we've got a game code. So I've got over 60 something review games already made for you. All you have to do is go to the Quizzes website and type in that code, and you'll be able to get these games already open, free to play, just meant to help you. Okay. So we got that. There we go. And then we also have FRQ Fridays. So last year I did 12 different FRQ Fridays. I went through FRQs. I explained to students like how you can find points, even if you don't know anything, like where you can pull information from the prompts. You can access my daily review. I'm posting the questions every single day. That's going on my Instagram is also being posted into these little folders. So if you click here, it'll open up a folder that has all of the questions in there. So you can review using that. And the beast of all beasts, there's a 316-page review guide that you are open access, can use whatever. It's got topic questions, it breaks down by the I can statements. It is going to allow you to be able to kind of work through what do you know, what do you still need help with. I've got multiple choice questions built in. I've got the response questions built in. It's a 316-page document that just, my beast. I call it a beast review guide. So it is available to you. Everything on this website is 100% open access, all that good stuff. So after saying all that, let's see, what kinds of stuff do we have in our chat? FRQs are not that bad. How come you wouldn't be able to make ATP from fermentation? You can make ATP from fermentation. You just don't make as much as you do with aerobic respiration. Oh, y'all already answered that. Many FRQs look scary because I have so many words. Yeah. So in terms of y'all's FRQs, you really need to be kind of take a moment and really see what the questions are asking you. A lot of times the questions are not as hard as you think they are. They're not asking you as deep of a thing as you think they are. So underline in your prompt, use your diagrams. Those diagrams oftentimes can give you 1 or 2 points. If you have a diagram, use it. On number 5, you're gonna have to draw something. If they tell you to draw something and you don't know, draw something anyways. Just try. The worst that can happen, you don't get it right. No harm, no foul. But if you don't even try, you're automatically not going to get that point. Okay. Also, if it says predict, increase, decrease, stay the same. Put a prediction. It pumped into the membrane. It didn't pump into the membrane. It moved. It didn't move. It had an increase. It had a decrease. Give them something. These readers, we want you to get the points. We're begging for you to get points. So give us something that we can read and try to get you some points, right? You got to at least try. If you don't try, what are you doing? I'm not. So in terms of multiple choice questions, that's kind of what we were going through before. We went over some of those multiple choice strategies. So underlining, using your diagram. There was a hour-long session that I did with Marco Learning at the beginning of, I think it was like in January, I may have done it. So definitely check out their channel. There is an entire session on multiple choice strategies, and I go through, I think, like 6 or 7 different multiple choice questions using those different strategies, and they're all redesigned questions that I'm able to kind of apply as much as possible to what you're going to see in May. You seem to struggle with multiple connections and you end up overthinking it. Yeah, overthinking is definitely something that happens. Multiple choice, you can always cover up your answers and see if you can come up with the answer on your own. Because if you came up with it, there's a big chance that that's the right answer. And before I forget to mention it, because I forgot last time, don't forget, follow Marco Learning on Instagram, follow me on Instagram, APBioPenguins, Marco Learning. We're both on TikTok. John's usually cutting up some random vegetables just to get y'all interested. But we do have a lot of good information to share with you, and everything that I give you is always free, just trying to help you out as much as possible. And of course, the YouTube channel where you can find all these videos as well as other videos. So I don't see any other questions in the chat. So I'm going to say, have a wonderful day. And remember, APBioPenguins are just a success.